Welcome to Biology Made Easy. In today's lesson, we are discussing the genetic code. It's the sequence of bases in DNA. Well, we have DNA double helix here. This is the sequence of bases. And the sequence of these bases determine the type of amino acids that should be put together to make one polypeptide. Adenine, thymine, cytosine, guanine, whatever the sequence is transcribed or copied into messenger RNA and the other RNAs and goes to determine the sequence of amino acids that form a polypeptide. The bases, they work in threes. So the genetic code is said to be a triplet code. 3 bases in DNA code for one amino acid. And we want to have a proof of that. There are 20 amino acids in nature that form proteins in nature. And the bases in DNA, there are 4. Adenine, thymine, cytosine, guanine, 4 bases. So, 20 amino acids in nature, there are 4 bases. If one base code for one amino acid, then there will only be 4 amino acids. And yet there are 20 amino acids. If 2 bases, each of these two code for one amino acid, we'll have 16 amino acids formed. If 3 bases code for one amino acid, we'll have 64 amino acids. So the genetic code have been found to be a triplet code. 3 bases in DNA code for one amino acid. This triplet code is also called a codon. So you meet a codon, it means triplet bases. So 3 bases in messenger RNA or DNA is a codon. So if we have 64 codons, all right, then the genetic code can be said to be a set of 64 different codons that code for amino acids. And yet it's been found that only 61 codons code for the 20 different amino acids. And 3 codons are stop codons. We'll talk about that soon. So we can say that the genetic code is a sequence of nitrogenous bases in the DNA that code for amino acids that should be included in a polypeptide. And that the genetic code is a set of 64 different codons. And the 64 different codons, 61 code for amino acids and 3 are stop codons. So we can bring this to add to our definition of the genetic code. The genetic code is also universal. It means that the same triplet bases or codons that codes for one amino acid in one organism is the same triplet basis or codon that will code for the same amino acid in all organisms. There are 20 amino acids that form proteins, and the same amino acids are in all organisms, and the same codons, triplets or bases, code for those same amino acids. Universal genetic code. Then the genetic code is redundant or degenerate. It means that many different codons stand for the same amino acid, or some amino acid are coded for by many codons. For instance, you have these codons. See, we have it in messenger RNAs, because the RNAs are the ones that are going to put in the amino acids together. So we have GGG, GCA, GGU, GGC. These different codons, they will all code for the inclusion of amino acid glycine in a polypeptide. Then you have U, UA, all these different codons code for the amino acid leucine. Say that amino acid leucine be part of the polypeptide. These also code for the same leucine, but then note here, UGG is a codon that codes for amino acid tryptophan, and that is the only code. Then you have a code AUG coding for amino acid methionine, and that's the only code. This AUG methionine is a start codon. This amino acid, we are going to look at what the start codon is. Well, so the genetic code is punctuated, meaning there are start and stop codons. It means that if this is a gene, this gene is made up of triplets. That's codons, triplets. These three bases stand for or code for the inclusion of an amino acid. These three, and there will be some codons that do not code for amino acid. So we have start and stop codons. At the onset of making or coding for a messenger RNA, there's a start codon, and that codon is AUG methionine. Look at it here, methionine is an amino acid. This methionine is a codon that starts the beginning of every messenger RNA, and that AUG will finally stand for methionine amino acid in a polypeptide chain. So there's always a start codon, and that start codon is an amino acid, methionine. And there's always a stop codon that will say stop putting in amino acids in this polypeptide. So for every polypeptide that is formed, the first codon is methionine, AUG. That's the start codon. And then the amino acid will be strung. And when the stop codon is reached, the stop codon will give the signal stop linking amino acids. And the stop codons, there are three types of stop codons: UAA, UAG, UGA. They are codons that do not code for amino acids. They only signal to stop adding amino acid, and they are referred to sometimes as nonsense codons. Then the genetic code is non-overlapping. It means the codons are read straight. For instance, if there's one strand of DNA, the genetic code is read ATG, that's one, CTG, one, CTT. So ATG, CTG, CTT. It's not ATG and then TGC and then GC. No, just straight ATG, CTG, non-overlapping. The same thing happens in RNAs. Messenger RNA, for instance, you have UAC, that's one codon, GAC. So the genetic code is non-overlapping. Well, this is what we have for today. If the lessons have been useful, please give us a thumb and kindly subscribe. Thank you. Goodbye.